translation. What is called as the translation? This is the protein factor. This is the unit into your protein metabolism. Translation and the transcription, it is very important topic to making down of the protein process. Okay, let us talk about the translation. The translation process of converting information stored in nucleic acid sequence into the protein. Means ki the nucleic acid, like this is the building block of the proteins, which are how it will be arranged into to making down a protein. This is called as the nucleic acid sequence into the protein. This is called as the translation. The sequencings of the mRNA, means ki messenger RNA, are translated into unique sequence of amino acids in a polypeptide chain according to the specifications of the genetic code. Means ki which is the sequence of the mRNA, means ki messenger RNA, which is translated into the unique sequence, means ki which are translated by into the unique factor. We are studied by the step by step how it will be arranged into some manner, that is the messenger RNA and amino acid in a polypeptide chain according for the specification of the genetic code, which are translated by the genetic code. Let's start. Which are take place, where it is take place, which is take place in the cytoplasm. So now you are aware about that the cytoplasm, other than the nucleus, that is the cytoplasm. Exceptions are few proteins coded by mitochondria and chloroplastic DNA, except some proteins which are made by the coded by the mitochondrial and the chloroplastic DNA, which is performed on ribosome, which is performed by the ribosome. There are large and the small subunit by the ribosomes. What is necessary for the translations? Like this is template mRNA, tRNA, which is transfer RNA, which is linked to the amino acid, ribosome, many accessory proteins, means ki some accessory related of the protein, and some energy by the GTP hydrolysis, means ki some energy sources which are necessary for the translation process. mRNA, the single stranded molecules of RNA which is encoded sequence of polypeptides, means ki which is single stranded molecules of the RNA which is encoded by the sequence of the polypeptide, transcribed and processed in the nucleus and then exported into the cytoplasm, means ki which is transcribed into the process by the nucleus, means ki which is translated factor which are occur, the all type of mechanism which is observed into the nucleus and transcribed into the cytoplasm, means ki exported into the cytoplasm. 5' end has binding site for the translation initiation process, which is translate by the 5' end has binding, the starting point which is from to the 5' and translated into the initiation process, means ki which are translated by into the initiation factors. Middle, which is encoding sequence, means ki the middle factor of the mRNA, which are the mechanism factor, which is sequencing factor, and 3' end, which is regulated by the stability of the mRNA, means ki the end of the 3' which are regulated by ability by the messenger RNA. Next one, the codes for one protein, means ki which are code, specific protein have the specific nucleotide sequence, means ki they have the some sequence factor which is capped on both 5' and the 3' end, means ki every have the 2' ending, 5' end and the 3' end. 5' cap, which is methylated cap, means ki the ending 5' cap which is made by the methylated cap. Protects from nucleus, means ki which is protected by the nucleus. The 3' poly A tail, which is made by the 3' poly A tail. Poly means ki a long chain of the poly A tail, and 50 to 200 adenines and added post transcriptionally, which is post means ki started before, this is called as the post transcriptionally. Protects mRNA from the degradation, means ki which are protected by mRNA situations before degraded factor. This is protect mRNA from degradations. Next one. Second point, what is called as the genetic code? What is called as the genetic code? The codons. First one, what is called as the codons? 3 nucleotides which are arranged by gather and making down of one protein. This is called as the codons. 3 nucleosides which are attached each other and making down of some protein. This is called as the codon. Codon, which is specific sequence of the amino acids, right? Which is initiation, which is by the start initiated codon. Why it's been need to genetic code? Because most of the things which are observed by the genetic code. Initiation means ki by the starts codon. AUG, like, so AUG, which is a code of the methionine. Every protein in a cell which is starts with the methionine, means ki every protein into the cells which starts by the methionine. And termination means ki stop codons, which are terminated by this process by the stop codons. Like, what are the stop codons? The 3 types of the stop codons are there: UAA, UGA and UAG. These 3 are the stop codons, right? So these are all required for the factors. See, these are the using of the genetic code. Why it's need to genetic code into the translation process. Next one, genetic code which is universally same, means ki we are only talking about the human being, so which is universally it is same, which is degenerate some amino acids are specified by more than one codon, means ki which is degenerate some amino, degenerate means ki dearrange, degenerate again and again, which are combination of some Punnett square mechanism, right? This is the some amino acids are specified by more than one codon. 64 possible codons and only 20 amino acid, means ki which are from to the 20 amino acid. Out of them, how much type of the possibilities of the codons are there? So there are 64 possibilities observed by the 20 amino acids. Next one, see, this is the genetic code. How it will be represented? This is the type, means ki these are the 20 amino acids. These all are the 20 amino acids which are there. There are 4 type of the factors here which are observed here, and this is UUU, UUC, UUA and UUG. This is how to be matching over there and making down of some possibilities. There are 4 possibilities of this type of the proteins which are outcome over there. I hope you understand how it will be make up of the amino acids by the protein factor. Okay. Next one. See here some process. This is the methionine, where the translation process which is started over there, which is observed in a yellow color and which is observed by the red color, which are called by the stop codons. Means ki any translation process or any mechanism we have to stop, we need by these 3 type of the codons required: UAA, UAG and UGA. These 3 type of the codons we are using for the termination process. Next one. Now, what is the role by the tRNA? Means ki transfer RNA to deliver amino acid to the translation complex, means ki which are transferred by the tRNA, means ki transfer to the amino acids. Serve as the adapters between codons in mRNA and amino acids, means ki which is serves as adapters between the codons by the mRNA and the amino acids. 4 steps and the 3 loops, means ki they have the contains 4 steps by the 3 loops. This is 1, 2 and 3. 1, 2 and 3, the major 3 loops which are observed. First one, anticodons by the decoding triplet localized on the anticodon stem, means ki which is the anticodon factor. Here by the anticodon factor, what the type of the antibodies, or sorry, what the type of the amino acids which are situated here, as according to here another type of the amino acids which are attached by in a way. So this is the attachment by the triplet localized on the anticodon strands, right, onto the ribosomal site, which consists of a single RNA strands that is only about 80 nucleotide long, means ki consist with the single RNA strand and that is only about the 80 nucleotide long, means ki how much long it is? It is long as the 80 nucleotide long. Each carries a specific amino acids on one end and has an anticodon on to the other end. Means ki each carriers which is specific amino acids on the one end and has anticodons on the other ends. A special group of enzymes pairs up the proper tRNA molecules with their corresponding the amino acid, means ki specific type of the groups of the enzymatic pair up by the proper tRNA molecules with their corresponding by the amino acid, which is correspond by the amino acid. tRNA brings the amino acids to the ribosomes, means ki the tRNA which is brings by the amino acids to the ribosome, which are initiated by the ribosomes, which is brings by the tRNA factor. Now, how the tRNA looks like? The tRNA which is looks by the L-shaped secondary structure, which is the L-shaped by the secondary structure, and anticodon and the amino acids are at the opposite arm of the L. Means ki see, this is the amino acid over here and this is the anticodon factor which is attached by this factor. This is the methyl related cap, observe here. Charging of the tRNA, so which is linking by the amino acid to correct tRNAs, which is charging, link by the amino acids by the correct to the tRNAs, which is catalyzed by amino acyl tRNA synthetase, means ki amino acid tRNA synthetase, couples an amino acid to its cognate tRNA, couples by the amino acid its cognate collected by the tRNA process, which is fidelity of the coupling 20 different synthesis. Fidelity means ki synthetic mechanism by the 20 different factors, which have the two steps, First one, activation of the amino acid and transfer of the amino acid to the tRNA. It consists by the two factors, two steps, activation of the amino acid and transfer of the amino acids to the tRNA. Next one, activations of amino acids by ATP means amino, acyl and AMP factor, which is activated by the energetic factor, means ATP in the formation of by the ATP by the energetic factor, which consists of the high energy intermediate, means in intermediate process, high energy process which are consist. Next one, transfer of the amino acid from amino acyl AMP to 3' end of the tRNA by the adenine, means ki amino acid how it will be transferred from the amino acyl AMP to 3' end of the tRNA, means ki tRNA by the adenine, which bonds are the covalent bond, which is bonding by the covalent bond formation. The high energy bond prepared by amino acids for the formation of the peptide bond with the nascent peptides, means ki high energy bond which is prepared by the amino acids for the formations of peptide bond, which is formed by the peptide bond with the nascent peptides. Nascent means ki a specific nascent peptide which are observed. Next one, this is most important, the ribosome organelles, which is most important organelles to making down of the translation process. The ribosomes, RNA or the protein complexes, so which is ribonucleoproteins, place of the translation by the protein synthesis, means ki which is the translation take place by into the translation and by the protein synthesis, which is abundant in a cell that synthesize large amount of the protein. Abundant means ki connected in a cell that synthesized by large amount of the proteins, means which is made by into the large amount of the proteins. Structurally and functionally similar among a species which is differ between prokaryotes and the eukaryote, means we are only talking about the eukaryotic, but in a prokaryotic and eukaryotic the genetic factors which are different, the genes code which are different structurally and functionally, similar by among species. Similar means there are same, but the coding factor which are different, right? Next one, how it will be initiated. See, this is the exit site, this is the peptide site, and this is the entry sites, right? This is P site, A site and E site. This is the ribosome. Here this is the small subunit, this is the large subunit. These three factors which is involved by the ribosome and translation process which are observed. Ribosome, which is composed of the small and the large subunit. The ribosome which are composed by the two factors, small and the large subunit. Whatever I told you that subunits means binds together for the translation. The ribosome self-assemble without additional factors, means a ribosome which is self-assemble without by the additional factors. This is the ATS which are observed into the eukaryotic factor. Now, here this is the 5 prime, here this is the 3 prime, this is the small subunit, this is the large subunit, which is decoding and synthesis takes place in the cavity between the subunit. This is the cavity between the ribosomes, and ribosome move along mRNA chain during the translation, means ribosome which are moved by the mRNA chain during for the translation process. New peptide exists through the tunnel in the large subunit, means the peptide which are exist through by the tunnel in the large subunits. These are the tunnel by the large subunits. Next one, this is the observed into the eukaryotic. See, this is the bifurcations which are observed by the eukaryotic. 40, 60, 80 subunit. They have the 80 Swartzberg factor. This is S is stand for the Swartzberg. This is the unit of the cell, means cell organelles. This is how much protein which are involved, 33 protein, and the 49 proteins which are involved by the 60S and the 40S. Small subunit which is contained by the 18S RNA and 33 protein. 18S RNA means how much amount of the RNA which are used, 18 Swartzberg, and 33 proteins which are involved to making down of the this 40S. Okay, next one. Ribosomes which is cytosolic, bound to the endoplasmic reticulum, which is also located into the mitochondria and chloroplast of the eukaryotic cell. How it will be observed? In a 3 factor, cytosolic, free, cytoplasmic factor by the factor means cytoplasm, obviously ribosome which is observed by the cytoplasm, due to of that reason the whole process which is making down into the cytoplasm, so it's called as the cytosolic factors, which is bound to the ER. Why? Because endoplasmic reticulum which is bound to the ribosomes which is made by the rough endoplasmic reticulum and generate some proteins. Simple way, which is also located into the mitochondria and chloroplast of the eukaryotic cell. Now, free ribosome which is found in the cytosol, which are found into the cytoplasm and may exist as a single ribosomes or in a groups known as the polysomes, which are occur in a greater number than bound ribosomes in a cell that retain most of their manufactured protein in the cytosol, means which are occur greater in a number bound to the ribosomes which are occur into, because the RNA which is the one type of the factory of the protein in the inverted comma. So we are talking about the ribosome style organelles here. Now, bound ribosome means bound to the exterior of the rough endoplasmic reticulum, means RER. This is rough endoplasmic reticulum which is generated by the specific proteins. which is occur in a greater number than the free ribosome in cell that secrete than manufactured protein. Means which is occurred in a great numbers than free ribosome in the cell which are secrete their manufactured protein, like pancreatic cells. These are manufactured. This is example over here, which is greater number made by the ribosome cell. Whatever type of the protein which are generated, translation process is there. Here we are talking about only the pancreatic cell. Okay, next one. Here some various factors which are involved during for the translation process. Means which is processed by the three factor: initiation, elongation and the termination, which is caused by three phases of the translation. Initiation means starting, elongation who are started which are going to be elongated, and termination means whatever transcribed, whatever translated which are going to be stopped, because the requirement of the protein which are going to be stop over there, that process which is the mechanism which is stop, this process called as the termination. Here some capital words over here. These are all the factors name which is specific responsible for the eukaryotic, which is specific responsible for the eukaryotic cell. And here the role over here, observe here. I am just taking this classification of this class for your just information. This is not asked in your examination. I am giving some extra knowledge about it. This is factor name, this is role name, and this is the process name. Next one. How to be protein synthesis means how to be protein making down. Synthesis means generated. So which is, as according to I told you that in previous slide, which is generated by three phases: initiation, elongation and the termination. First one, initiation. How the initiation process which is going to be there. Initiated means started. Attachment of the initiator tRNA means methionine-related tRNA to start by the codons on mRNA, and assembly on ribosomal subunit. Assembly means gathering onto the ribosomal subunit. First of all, tRNA by the methionine-related cap from to this is initiated, and mRNA which are going to be assemblated. mRNA situation which is assemblated over there, methionine-related cap which are observed there, and the ribosomal subunit which are going to be gathered at one side. These are the generating factors. Next one. Several initiation factors assist to the process. Means several type of the initiation factors which are assist by this process. What is that? Strongly which is regulated process, which are strongly by this regulated process. This is the regulated water then arrangement by of the regulatory factor. Uses energy from the ATP or GTP hydrolyze, which are using by the high energy factor ATP or the GTP factor. Which is cap recognition. What are the cap recognition? Methionine-related cap recognition. mRNA initiator factor means mRNA which is initiation by this factor. Helicase means ATP is unwind to the mRNA. Formation of the pre-initiation complex, which is the formation. How to be formation? The pre-initiation factor what is required, which is made by the first initiator by the tRNA, means which is a tRNA, where it is bounded, where the methionine-related tRNA which are observed, that side to the tRNA first binding over there by the anticodons. Now we are aware about the anticodons factor, and bind to the small subunit of the ribosome, which is bind by the small subunit of the ribosome. Initiation factors which is used by the GTP hydrolysis, means the initiation factors which are used by the GTP hydrolysis process. Unwind and scanning, which is by the unwind and scanning by the mRNA process by the methionine cap by the ribosome. Complex scan, here some complex which are scanned and binds to the start codons, which are bind by the start codons. Dissociations of initiation factors. Association with the large ribosomal subunit, associate with the large subunit by initiation factors which are IF1, IF2 and IF3. IF means initiation factor 1. This is in a capital letter. Be remember there, which is capital letters which is represent as the eukaryotic translation process and the genetic code, who gives some information, give some informations to the cells and making down of all the mechanism by this way. Next one, what is the elongination? Means repetitive cycles, means ki whatever occur into initiation process which is going to be repeated by, because we are required major type of the protein, means ki we are required till to the requirement of the protein is not completed, till to the elongation cycles would be occur. Repetitive cycles of the codons, directed addition of the amino acid tRNA, which is the addition of the mRNA tRNA, amino acid tRNA binding, which is by the mRNA, right? Amino acid tRNA binding, anticodon factor, proofreading by the large and the small subunit in the cavity portion, by the A site, P site and E site. Peptide transfer, means ki protein will be transferred by to the A site, P site and the exit site. Translocations, means ki which is translated by the another factor. What are the elongation factor? EFTU, EFTS and EFG. These are C, elongation factor, TU, elongation factor, TS, elongation factor G. These we are elongated into a process which are repeatedly occur. Elongation, amino acid tRNA binding, elongation factor, small EF, which is derived amino acid tRNA to the ribosome, and the G which is bound to the GTP. This is the energetic factor, which are a higher energy factor which are required for the elongation process. Next one, proofreading. What is called as the proofreading? Between of the small subunit and large subunit, whatever type of the protein or the materials which are generated exactly like of the requested mRNA, which is attached by the methylated cap, exactly that type of the generated or synthetic protein which are observed, that is called as the proofreading, right? That is mRNA DNA check against by the codons, only attached by the codons, that by the sites. GTP hydrolysis and dissociations of the EEF TS from the ribosome. Elongation site of the TS from by the ribosome. Correct amino acid tRNA retained based on the codon anticodon pairing, means ki tRNA which is bound on the mRNA, and these all are gathered into the ribosomal subunit cavity, and whatever type of the mRNA sequence which is observed, tRNA which is attached by that anticodon sequence and gives some protein materials over there by the tRNA. Are you getting my point? Incorrect amino acids tRNA escape from to the ribosome. If the anticodon factor of the tRNA which are observed, which are escaped by this process. Translocation, which is translocation means ki one side to the another side. Binding by the EFG GTP. Movement of the peptidyl tRNA to the P site. Sliding from the mRNA by the 3 bases. tRNA which is moved to the exit, and which is the translocation site, A site, P site and E site. A site, P site and E site. GTP hydrolysis and released by the EFG. Now the last process, termination. The cycles which are going to be stopped thereby because no required of the next for the generated synthetic protein which are required. This is the end, means ki stop down of that mechanism. This is called as the termination. The final step in translation, which is termination, when the ribosome reaches by the stop codons, which is whatever I observed in your table, that is the genetic code. This is the 3 type of the stop codons which are now taking their in action, means ki release codons, stop codons, nonsense codons, which are correspond by the tRNA. Instead, a small protein called as the release factor attaches by the stop codons. Then the release factor which is causes the whole complex to fall apart like mRNA and 2 ribosomes by the subunits. the new polypeptide and the new polypeptide which are going to be there. The messenger RNA can be translated many times to produce many proteins copies, means ki the translator factor which are made by the factors, many protein copies which are there. Termination factors which are RF1, RF2 and RF3. These three are the termination factor, termination genes which are observed.